So this video is for Chapter 2, and we are only going to cover two sections of Chapter 2, Section 213 and 214. So this chapter primarily contains a lot of stuff that is covered in intro biology and intro chemistry courses like atoms, types of chemical bonds, basic energy concepts, enzymes, properties of water and aqueous solutions, pH, and macromolecules, including carbohydrates, lipids, and proteins. And we are only going to cover the sections on nucleic acids and ATP because they are the most relevant to future chapters. So first, section 213, which covers the nucleic acids, DNA, and RNA. So nucleic acids are macromolecules, and a macromolecule is a large molecule called a polymer, which is actually made up of repeating units of smaller subunits called monomers. So nucleic acids are macromolecules containing carbon, hydrogen, oxygen, nitrogen, and phosphorus. They function to store, transfer, and process information and instructions. So our nucleic acids are holding all of the information that allows our bodies to grow and develop into human organisms. There are two types of nucleic acids, DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. The structure of a nucleic acid is that there are usually one or two long chains of monomers, and again, the monomers are the subunits that make up a polymer, and these monomers or subunits are called nucleotides. So RNA is usually single-stranded, so one strand is shown here on the left. DNA is usually two-stranded or double-stranded as shown on the right. So a nucleotide, again, this is the monomer or subunit of a nucleic acid, and there are three components. There is a pentose sugar, which is shown by the little light gray pentagon here. And this sugar is either ribose in RNA or deoxyribose in DNA. There is also a phosphate group that is attached to the sugar. And then there is a nitrogenous base. That means it is a compound that contains nitrogen. And there are 5 bases: adenine, guanine, cytosine, thymine, and uracil, abbreviated A, C, G, T, and U. And so again, a nucleotide has 3 parts: a sugar, a phosphate group, and a nitrogenous base. Now, when these monomers come together to form a polymer, the sugar and the phosphate group are the part that will make up the backbone of the polymer, and the latter rungs that stick out to the side are made up of the nitrogenous bases. So again, looking at the different nucleotides, purines, which are adenine and guanine, the A and the G, purines have 2 rings. So again, you see the nitrogens in there, that's why they're called nitrogenous bases, and they have 2 carbon rings is what makes them a purine. So adenine shown in red, guanine shown in green. Pyrimidines have just 1 nitrogenous ring, and the pyrimidines include cytosine, represented by C, thymine, represented by T, and uracil, represented by U. And then notice that thymine is only found in DNA, and uracil is only found in RNA. So again, if we look at the structure of the nucleic acid, which is the polymer, it's the macromolecule made up of the multiple repeating subunits, then we can see the sugar-phosphate backbone, which is shown here in the RNA molecule. Remember, RNA is usually single-stranded. And then over here in the DNA molecule that is double-stranded, the sugar-phosphate backbone is the actual outside ribbons. And again, the sugar-phosphate backbone is made up of the sugar group and the phosphate group. And then the bases, the nitrogenous bases, stick out to the side. So again, in our single-stranded RNA, we can see them sticking out to the side. And then with the double-stranded DNA, The bases not only stick out to the side, but they match up with bases on the other strand of DNA, and they do so in what's called a complementary fashion. So the A will always want to pair up with a T, or in the case of when DNA is interacting with RNA, the A in DNA will pair up with a U in RNA, and the C will always pair up with a G. So that's what we mean by complementary bases. So here's a chart to compare and contrast DNA and RNA. So RNA, the sugar is ribose. DNA, the sugar is deoxyribose. They both have adenine, they both have guanine, they both have cytosine. Ribose has uracil as one of its pyrimidines, and DNA has thymine as one of its pyrimidines. The number of nucleotides, remember the nucleotide is a subunit, that is found in the overall nucleic acid or polymer. In RNA, it can vary from fewer than 100 nucleotides to around about 50,000. DNA is very large. DNA can have like more than 45 million nucleotides in one DNA molecule. With RNA, the shape can vary. So they're usually single-stranded, but that single strand can fold itself into many different types of shapes. And we're going to talk about the 3 types in just a minute. DNA is always paired strands that are coiled together into a double helix. RNA functions to make proteins. So DNA actually has the instructions and it passes the instructions to RNA, and RNA does the making or the synthesis of the proteins. DNA stores the genetic information, so it's the master repository of all of the information needed to make those proteins. So there are 3 types of RNA, or 3 main types. There are actually more than 3, but there are 3 main types that we are very familiar with. There is messenger RNA, abbreviated mRNA. There's transfer RNA, abbreviated tRNA. And there is ribosomal RNA, abbreviated rRNA. They are all 3 involved in protein synthesis, and we are going to talk briefly about protein synthesis when we get to chapter 3 and talk about the major functions of a cell. But typically the messenger RNA is carrying the message from DNA out into the cell. The transfer RNA is helping to transfer amino acids in to make the protein, and the ribosomal RNA actually forms the structure that makes the protein. So to sort of summarize nucleic acids, DNA contains all of the master instructions on how to make all of the molecules needed for the cell to survive and live. And remember, cells make up tissues, tissues make up organs, organs make up organ systems make up organisms. So the instructions for everything ultimately boil all the way back to DNA. And the RNA takes those instructions provided by DNA and uses them to make proteins. And as we'll see in chapter 3, proteins do the majority of the jobs in a cell. So ultimately, DNA is providing the instructions, RNA is building the tools, using those instructions, and then the tools go out and do the jobs. And the only other section we're going to cover in chapter 2 is the section on ATP. So I'm going to remind you that the cell is the smallest living unit that makes up the human body. Cells make up tissues, tissues make up organs, organs make up organ systems, and organ systems are essential to the overall homeostatic dynamic equilibrium of the organism. So it all boils down to all of the little billions of cells in our body each one has their own job, and they all have to do those jobs by using energy. And so for energy, cells use these compounds called high energy compounds. And these are compounds that have phosphate groups attached to them. And when a phosphate group is attached to these compounds, that is considered to be a high energy bond. So the method of phosphorylation is when a phosphate group gets attached to a molecule. So you can take phosphate groups off by dephosphorylation, or you can stick them on by phosphorylation. So basically what this means is if you have some molecule in your body, shown here like by an orange circle, for example, we can come along and stick a phosphate group on it, and we'll talk a bit about what's going on here in the middle in just a minute, but we can stick a phosphate group on it and notice how it can change the shape of the molecule. So that's one way that phosphate groups can cause molecules to do their jobs, is by changing their shapes. In the cases of like enzymes, which are proteins that actually carry out reactions in our cells, phosphate groups can sometimes turn them on. So if the enzyme doesn't have the phosphate group, it's not working. You stick a phosphate group on, now the enzyme is working. In other cases, the enzyme is working if it doesn't have a phosphate group on it, and then you can stick a phosphate group to turn it off. The bottom line is that the transfer of phosphate groups can make things change inside the cell, which helps the cell get its jobs done. So just like some people say, well, I can't get going in the morning until I have my cup of coffee, there are some enzymes and other molecules in the cell that say, well, I can't do my job until I get my phosphate. So the main energy currency of cells is adenosine triphosphate, which we abbreviate ATP. So this is the main molecule that is used as energy currency in actually most living systems. So ATP, or adenosine triphosphate, has a very similar structure to adenosine, which we looked at earlier. It was one of the nucleotides. But if you remember, a nucleotide has one phosphate group, a sugar, and a nitrogenous base, but adenosine triphosphate, as the name suggests, tri means three, has three phosphate groups. So again, it looks just like a nucleotide, where we have adenine, which is the nitrogenous base. We have the ribose sugar. And whereas a nucleotide only has one phosphate, ATP has three phosphates. And each of these is considered to be a high energy bond. So what we can do with ATP is we can take those phosphates away from it and put them on other molecules to wake those molecules up and have them do the jobs they are supposed to do. So while all three phosphates are present, we call this molecule adenosine triphosphate, or ATP. We can take one phosphate away, and if we only have two phosphates, we call it adenosine diphosphate, or ADP. And in some cases, we can take two away and we're left with one, in which case we have adenosine monophosphate, abbreviated AMP. So as I've already said a couple of times, in the body, in cells, energy is released or stored by removing and adding phosphate groups. So here is ATP at the top. And again, we have our adenine, our ribose, and our three phosphates. So if we take a phosphate away, right, we can use that phosphate to do work. So we can stick it on an enzyme, wake it up, tell it to do its job, right? So that phosphate represents work being done by something in the cell. And of course, since we took a phosphate away, we are left with only two phosphates, so we are left with ADP. Now conversely, if we have extra energy lying around, so let's say you just ate a big dinner and you've digested that dinner and you've absorbed a lot of the nutrients, now you want to store some extra energy for later when you go on a run. Now you can take that extra energy in the form of a phosphate, you can stick it onto ADP and make a new ATP that then you can use in the future to get jobs done. So to review in equation format, and you should know both these equations, if we take ATP and we break off a phosphate, that is releasing energy. So if we break off a phosphate, we get energy that we can use to do something, and we're left with ADP. And then if we have extra energy to store, we take the ADP, think of it as like the empty molecule, we add the phosphate back to it along with the energy we want to store, and it's now stored up inside ATP. So think of ATP as money in the bank, right? It's ready, it's there to be used, and ADP is the empty bank account, right? So you can add the money to ADP to store it in the form of ATP, or you can take the money out to use it. There are some other high energy compounds, guanosine triphosphate and uridine triphosphate. So ATP is not the only one, but ATP is the major one and the one that we're going to mention several times throughout the remainder of the semester. And that is it for Chapter 2. So happy studying. I do want to point out, because Chapter 2 is so short, I did not make an in-class worksheet or an in-class activity for Chapter 2. Instead, the concepts of the nucleic acids and ATP are going to be built into the Chapter 3 activity and the Chapter 3 worksheet.